Today, we will talk about cell-cell communication. We'll start by going over an overview of what this means and generally how the process works. Then we will focus in on 5 different mechanisms by which cell-cell communication can occur. We will talk a little bit about the role of phosphorylation and dephosphorylation in allowing signals to be sent and understood by cells. We will talk about possible locations of receptors either in the plasma membrane or in the cytoplasm, and which properties of ligands will correspond to each of these receptor locations. Then we will talk about intracellular receptors, especially steroid receptors. And we will end by talking about membrane receptors. Let's keep in mind throughout this whole thing, the goal of cell-cell communication is to allow one cell to send a signal that will affect the actions of other cells. Given that many organisms are multicellular, then for that organism to be able to efficiently survive and accomplish its goals, its cells need to work in a coordinated manner. And this means that the cells need to be able to communicate with one another. When we have cell-cell communication, we will need 2 items to allow this to occur. First, we are going to need some signaling molecule, and we are going to call that a ligand. And on this diagram down here, you can see the ligand is this set of 2 green balls outside of the cell in the external environment. We then need something to receive the signal on the cell that gets it. This will be our receptor protein. And here that's represented by this blue shape sitting inside the plasma membrane. Finally, when the ligand attaches to the receptor, then we need some way for a signal to be sent. So once it is attached here, something is going to need to happen inside the cytoplasm so the cell can respond appropriately to the ligand. And we should specify that there are many different kinds of ligands and many different kinds of receptors, and they do not all interact with one another. Instead, a given ligand is going to interact with only one or a very small number of kinds of receptors. And the specificity of this interaction is based on the similarity in shape of part of the ligand to part of the receptor. So this should remind you very much of what we already said about enzymes, when we said that enzymes interact with substrates based on having an appropriate shape that allows them to fit together. The same thing is true here. A ligand that has the appropriate shape can fit into a particular location on the receptor, and once this has happened as sketched here, then there can be an intracellular response. However, if the ligand has a different shape and it cannot interact with the receptor, then no response would occur. Let's go back to a situation where the ligand in fact does fit with the receptor and talk about what will happen. There needs to be some way for the signal from that ligand to be communicated inside the cell. And again, this will remind you very much of what happens with enzymes. When a ligand binds to a receptor, it is going to have subtle effects on the shape of that receptor. So for example, here, we might have a receptor without its ligand attached on the left. Once a ligand attaches to it, for example, right here, then this will lead to at least a subtle change in shape. And the key here is the change in shape happens inside the membrane. So the ligand here is attaching outside, but this leads to a modification of the shape inside the membrane. So you can see that this red tail, which had been sticking off at this angle, now is at a slightly different angle. Well, even a small change like this can lead to big differences in what this protein does. For example, this change in shape could make an enzyme go from being turned off to turned on, or it could allow some other protein to be able to dock or attach to it, when in the other conformation, the additional protein would be unable to dock. That is the basic idea. That is the basic idea of how signals can be sent from cell to cell. What we see in animals is that there are signals that last for very different amounts of time and that send signals across very different distances. And we can divide then the kinds of signaling into 5 different categories based largely on the duration the signal lasts and how long that signal or how far that signal gets sent. And so we will talk about each of these 5 different mechanisms of signaling here. I'll give you a brief list to start with. We have direct contact between cells. Then we have autocrine signaling down at the end of this list, paracrine, endocrine, and synaptic signaling. We'll talk about each of those in some detail. First is direct contact. This is when there are 2 cells that are literally touching one another. And in this case, molecules on the surface of one cell can contact proteins in the plasma membrane of the neighboring cell. There are 2 kinds of molecules that often will be involved in these interactions. The molecules carrying the signal are usually going to be glycoproteins or glycolipids. We talked about these when we talked about the membrane about 4 chapters earlier. And you can see in this diagram a glycoprotein, this blue sort of feathery structure, and glycolipids, which are sitting in the membrane, but the sugar oligosaccharides are sticking out here. Remember that these are the signals. The neighboring cell would then have some receptors that are set up to interact with that signal or the signals on the glycoproteins. So the signals are recognized by receptors on the adjacent cell. Related to this is that some cells are actually connected to one another through openings. And we talked about these openings already. Specifically, we've talked about 2 kinds of openings, gap junctions. Remember, these occur in animal cells, and they connect the cytoplasm only. And second, plasmodesmata occur in plant cells, and remember, they connect both the cytoplasm and the endoplasmic reticulum. So that is an easy way for a signal to be sent if molecules can simply move from the cytoplasm of one cell through an opening into the cytoplasm of a neighboring cell. We'll move on now to autocrine signaling, and this gets only a brief mention in your book, but it happens, and so you need to be familiar with it. This is an unusual kind of signaling where a cell is sending a message to itself. However, instead of this message remaining inside the cytoplasm the whole time, the cell actually transmits the message outside into the extracellular environment. That signal then diffuses a very short distance back to a receptor on that own cell's plasma membrane. When the signal is received, that then leads to some sort of action inside the cell. This is an inefficient way of doing things, but signaling like this can evolve from paracrine signaling. You can imagine that initially this signal might have been sent to neighboring cells, but then something changed and it became useful for this cell to receive its own signal, and so signaling to itself developed as a mechanism. So that is autocrine signaling. Let's compare that to paracrine signaling, which we just mentioned. This is when a signal gets released from a cell and it affects nearby neighboring cells. This kind of signaling happens especially early in development when neighboring cells are going through the process of determining which cells will develop into one type of cell and which cells will develop into another type. And so by these cells signaling to each other, they can say, I'm already developing into some type and you need to develop into a different type of cell. Or conversely, they can say, we're all developing into the same kind of cell, you need to develop into this kind of cell as well. So paracrine signaling is going to allow clusters of cells that are near each other to coordinate their development. That's not the only time it's used. You'll see some other examples as well. A key to paracrine signaling is that signals are always going to nearby cells. A corollary of this is that the signal cannot last very long, because if the signals persisted for a long period of time, then by diffusion, they could diffuse further and further away until they would be received by cells that were not nearby or neighboring cells. So these signals must be short-lived. That means that these ligands are going to stop functioning fairly soon after they are released into the extracellular environment. Another kind of signaling is endocrine signaling. If paracrine was short-distance signaling and the signals did not last for long, endocrine is the opposite. In endocrine signaling, the signals are going to last for a long period of time, and that means they're going to have opportunity to travel long distances through the cell. In animals especially, this means that the signal is going to travel through a circulatory system. For example, through the blood from one area of the body to a distant area. Endocrine signaling can also happen in plants. For example, if a signal gets sent from a root to a leaf or from a leaf to a flower. And so these signals are long-lived and they travel long distances. Our next kind of signaling is going to be a signal that travels short distances, like paracrine signaling, but it is used only in one specific kind of situation, and the distances traveled are very, very short, even compared to paracrine signaling. This will be synaptic signaling. And synaptic signaling occurs between nerve cells or between nerve and muscle cells. This is when nerve cells release a neurotransmitter, so the ligand in this case is called a neurotransmitter, and upon release, that neurotransmitter is going to travel only a very short distance to a neighboring cell. For example, here's a nerve cell, and a signal goes inside the cytoplasm along the length of this nerve cell. But then this nerve cell needs to send the signal to a neighboring cell. This could be another nerve cell or perhaps a muscle cell. To do this, a ligand is released, but because these two cells are permanently very close to one another, the signal does not have to go very far, just across this short gap. And this neighboring cell has receptors that are set up to be in the right location to be very near the original nerve cell. So the signal can be received, and then the signal can be acted upon either by the nerve or the muscle. So this specialized arrangement where a lot of transmitters are available to be sent, or a lot of ligands I should say, and a lot of receptors are available nearby on a neighboring cell, this arrangement is called a synapse. And we will use the word synapse only for synaptic signaling involving nerve cells. As we've already said, these signals are very, very short-lived, and they travel a tiny distance, much shorter than paracrine. That then summarizes the 5 different mechanisms of signaling, which go a range of different distances. Next, we should talk about the fact that the same signal can lead to different responses in different types of cells. And there's a reason for this. Basically what this is saying is that the same ligand can elicit a different response in different cell types. In all of these cases then, the receiving cells will receive exactly the same ligand, but they won't do the same thing. The reason this can happen is that different kinds of cells have different response proteins inside, or sometimes, related to this, they have different co-activators inside. And so upon receiving the ligand, the receptor is going to be turned on in a very similar manner between these different cell types. But when the receptor is turned on, it's going to have a different effect because the response proteins it activates are different, or because the co-activators are different. We'll talk about co-activators when we talk more about steroid receptors. Let's talk about an example where different cell types have different responses to the same ligand. The example we'll use was covered in the book, and this is epinephrine. And epinephrine is another name for adrenaline. So this is a hormone released when one is scared or surprised. And if one is scared or surprised, then the body needs to do several different things. For example, it will need more energy sources. It will need to be able to move blood through the circulatory system faster. And the body will want to shut down some processes that are less important at the moment. All of these are included in the fight or flight response. So the flight or fight response is when your body says, something's wrong, I either need to get out of here quickly, or I need to tackle the situation at hand. So in this situation, the liver would want to release additional glucose. As you know, glucose is going to power respiration and allow things like your muscle cells to be able to fire faster. It is going to lead your heart to have stronger contractions, so the circulatory system acts faster. And it's going to make your blood vessels in some parts of the body contract and in other parts widen. For example, around the digestive system, they might contract because digestion is not important in the short term, but going to muscles, and let's say in your legs, they may widen so your leg muscles can get more oxygen so you can run away faster. So we're seeing three different responses here in different parts of the body, all from the same ligand. And what we'll see is that this can happen because each of these cell types have different response proteins inside that are going to lead them to be doing different things when epinephrine is received. We've talked generally about how a receptor leads to a response, saying that there's a change in shape. What we are going to see is that oftentimes receptors have their effect by causing a protein inside the cell to become phosphorylated, or less commonly, dephosphorylated. You're already familiar with the idea of phosphorylation. We've seen this when ADP is made into ATP, or when various sugars during, let's say, glycolysis acquire a phosphate group. That is phosphorylation. So phosphorylation simply means acquiring phosphate, and phosphorylation will be important in signal cascades because phosphorylation oftentimes turns on enzymes or sometimes turns them off. So phosphorylation is always going to occur by the action of a particular enzyme. Enzymes that add phosphate groups to a protein are called kinases. So a protein kinase will be an enzyme that adds a phosphate group to a protein. Conversely, a phosphatase is an enzyme that removes a phosphate group from a protein. You should notice in passing that both of these words end with ASE. As we've said earlier in the course, any word ending with ASE in biology means that the molecule is acting as an enzyme. Let's look at this on a diagram. Over on the left here, we have some protein, probably an enzyme, and it has a couple of particular amino acids on it. In particular, perhaps at this location, it has either serine or threonine. These are both amino acids that can receive a phosphate group. We then see that going from left to right, this amino acid goes from not having the additional phosphate group to having this phosphate group added on. If this has happened, then the enzyme or the protein has been phosphorylated. That means the enzyme that did this phosphorylation must have been a kinase. Conversely, going in the other direction, we can see that this phosphate group is removed, and so it becomes inorganic phosphate, or PI again. If this is the case, then it must be a phosphatase that is doing so. Now, going back to the original direction up here, note that this was not an inorganic phosphate that got added to our protein. Instead, the phosphate group was donated by ATP. This is because adding the phosphate group requires energy, and the inorganic phosphate does not have enough energy to be added. However, as you already know, that third phosphate group on ATP is associated with extra energy, and so removing it from ATP provides the energy that allows it to be attached to this enzyme. So we talked about the fact that ATP production is really important in cells. This is just one of the many reasons why it is. We can talk about a couple of different locations, in fact, just 2 locations where receptors can be on the cell receiving a signal. Some kind of receptors are going to be inside the cytoplasm, as pictured here. Remember, the prefix for inside is intra, and so these will be called intracellular receptors. If a receptor is intracellular, then logically, the ligand that turns it on, remember this ligand must be coming from another cell. So if that's the case, then this ligand must be able to get through the plasma membrane in order to activate this internal receptor. We will think a little bit about what must be the properties of this ligand if it's able to get through this membrane. Conversely, other kinds of receptors sit in the plasma membrane. Receptors sitting in the plasma membrane we can call cell surface receptors. And these can bind to ligands that never cross the plasma membrane. So here the ligand is outside, it attaches outside, and eventually it will be released back into the outside environment. These ligands then do not need to be able to cross the plasma membrane. We have already talked in other parts of the course about the properties of molecules that can cross a plasma membrane and the properties of molecules that cannot. So when you consider the properties of these, consider let's assume both of these molecules are relatively small. Think about which one must be polar to get across the membrane and which one can be nonpolar and stay outside of the cell without having to cross through the phospholipid tails. So it shouldn't surprise you then that steroid receptors, let's specify that steroid receptors are intracellular receptors. If they are intracellular, then that must mean that the ligand for them, the steroids, are going to be small and they must be nonpolar. Because if they were polar, they wouldn't be able to get through this region of the plasma membrane. So in this case, the ligand can cross the plasma membrane and bind to a receptor. Let's look at this and follow what happens. So here we have ligands that are crossing the plasma membrane and coming to bind to our steroid receptor. When this happens, we are going to see a few different events. And incidentally, you should be familiar with this pathway. So first the ligand crosses and binds to the receptor. Now notice here in turquoise, there's this inhibitor. What the inhibitor is doing is blocking active sites on the steroid receptor and preventing it from being active. When the ligand binds, this is going to cause a change in the shape of the steroid receptor that is going to make it lose its affinity for the inhibitor. Once it no longer has affinity for this, the inhibitor will be released, and now you can see the steroid receptor, or the hormone receptor, changes shape, and it, along with the ligand, are going to move through a nuclear pore into the nucleus. We now have the hormone and receptor complex inside the nucleus. Remember that our DNA is located in the nucleus, and the DNA contains the information to make proteins that will then be made back out in the cytoplasm. However, for the DNA to make proteins, then we'll talk about the details of this later in the semester, but some enzymes inside the nucleus have to be turned on, and they have to be alerted to exactly which genes to make proteins from in the DNA. And so without getting into the details of that, what you need to know for now is that when the hormone receptor complex attaches to the DNA, it has the possibility of turning on a process that is going to eventually lead to proteins being made back out in the cytoplasm. Now, we said earlier that different cells can have different responses to the same ligand. Let's see how that works specifically for steroids. We've seen that the ligand attaches to the receptor. This is going to be true in all cell types that have a receptor for this particular ligand. So thus far, everything would be identical regardless of where in the body this cell was. And this will move into the nucleus in a similar manner, regardless of where in the body this is taking place. However, the important thing here is that we need not only the hormone receptor complex to turn on this process of gene transcription, but we also need co-activators. So co-activators are other proteins that will interact with both the DNA and this hormone receptor complex. And it's the interaction of all three of those parts that are going to lead to this process that eventually leads to proteins. What this means is that if different types of cells have different co-activators, then the same steroid receptor complex can activate one set of genes in one cell, but it can activate an entirely different set of genes in another cell. So two cells of different types can make different kinds of proteins, even though the ligand is the same, and even though the hormone receptor is the same in both cases. We'll just define co-activators here as proteins that enhance gene expression. Gene expression is going to lead to protein production. And they're going to do this by binding to an activator. And in this case, our activator is the hormone receptor complex. The last topic that we are going to talk about today is 3 different ways that membrane-bound receptors can lead to a signal inside the cell. So remember, the steroids we just talked about were intracellular receptors, but now we're going to look at those that are stuck in the plasma membrane. Anything that is in the plasma membrane must have some hydrophilic regions on the outside and some hydrophobic regions neighboring these phospholipid tails. That means that this receptor cannot leave the plasma membrane and go travel somewhere inside the cytoplasm. It is stuck here. So if it is going to lead to some sort of action inside the cell, it is going to need to pass that signal on to some other molecule inside the cell that then has the freedom to move. This can happen in, as I said, about 3 different ways. The first way that we'll look at are chemically gated ion channels. So earlier in the semester, when we talked about plasma membranes, we said that there can be channels. We said channels were passive, they did not require ATP, and they were openings that allowed through usually one specific type of molecule, which is usually an ion. So this ion might be calcium, or it might be magnesium, or some other, oftentimes at least, cation. How these work are fairly straightforward. Basically, a ligand, or in this case, two ligands, will bind to either side of this channel. When that has opened, it's going to cause the channel to open and allow these ions to pass through, and so they could move towards their equilibrium as long as this is open. Eventually, these ligands will disconnect, and when they disconnect, then the channel will resume its closed shape, and the path for the ions to move will be closed off. So that is a chemically gated ion channel. Next, we can have enzymatic receptors. For an enzymatic receptor, the receptor might be turned off until the ligand binds, and once the ligand binds, even though it's binding on the outside of the cell, in the extracellular space, this can then lead to enzymatic activity of the portion of this protein that is inside the cell. So it goes from being inactive without the ligand to active with the ligand. Once it's active, then this can do something that then leads to a cellular response. As we said, one thing that often happens in signal transduction is phosphorylation. So perhaps in its inactive state, this cannot phosphorylate other proteins, but once turned on, then this enzymatic receptor will be able to cause phosphorylation of other proteins. And by doing so, some of those proteins might be enzymes that themselves get turned on by phosphorylation. And so that can then lead an inactive enzyme inside the cytoplasm to become an active enzyme and to go on and do something else. So enzymatic receptors are our second category of membrane-bound receptors. The third category is by far the most complicated, and these are G-protein coupled receptors, or GPCRs for short. And a GPCR has multiple pieces to it. First, let's look at the GPCR itself. It is a protein with, you can see, alpha helices that go through the plasma membrane. It doesn't just have one of these, but it has several of them. This looks kind of like a bunch of coiled snakes, and so sometimes you will see GPCRs called serpentine receptors. Serpentine because they look snake-like. Of course, there's still a binding site for the ligand somewhere outside in the extracellular space. Then inside the cytoplasm attached to the GPCR is a collection of 3 different proteins, and these are called alpha, beta, and gamma. When a ligand attaches to a GPCR, then these 3 proteins are released. It turns out that beta and gamma stay attached to each other, so they act as one unit, and alpha detaches and acts as its own unit. Now, what beta, gamma, and what alpha do varies widely for different GPCRs. So in some cases, beta, gamma go and attach to other enzymes or other channels and open or close those channels or turn on or turn off other enzymes. In other cases, it's the alpha protein that goes somewhere and activates or deactivates another protein. In some cases, beta, gamma go off and do one thing, while alpha goes off and does something else. And so I'm going to move this picture over to make it easier to see. Here you can see beta, gamma are activating a ion channel. Meanwhile, alpha goes off over here and activates another enzyme. When this ligand eventually gets released, then beta gamma are going to reattach to alpha, and alpha beta gamma are going to redock attached to the GPCR, effectively meaning that the GPCR is turned off until another ligand binds. We will talk about receptor kinases as part of our next lecture. To summarize a few important points from the material we've covered today, first, I want you to remember that different kinds of signaling relate to different goals. So think about the goals of synaptic signaling, which is to send a message very quickly, always to the same recipient cell, compared to paracrine signaling, which is to send a signal short distances to neighboring cells, compared to endocrine signaling, which has a goal of sending a signal a long distance throughout the body. And think about how the different ways that those signals get sent relate to what they do. Second, remember that the properties of ligands relate to the location of the receptor. So we've seen some membrane-bound receptors and some cytoplasm receptors, and we've seen that they have different properties for their respective ligands. And third, keep in mind that one signal can lead to different responses. And we've seen this in the steroid case because there can be different coactivators in the nucleus of different cell types. We can also see this in the case of things like enzymatic receptors, or in the case of GPCRs. To use GPCRs as an example, you could imagine that the same kind of ligand will be received by many different cell types, and those different cell types receiving the signal can have exactly the same GPCR with the same beta gamma and the same alpha. But if those different cell types have different enzymes inside, then what gets turned on by alpha in one cell type might be different than what gets turned on by alpha in a different cell type. So this means that the same signal, the same ligand, can lead to different responses because the response proteins in different cell types are different.